continuing our discussion about membrane transports, we need to look at active transports. Now, active transport can be used for several different things, some of which are going against concentration gradients. So normally, diffusion goes high to low concentration. But if you're trying to go from low to a high concentration, that could be active transport. You're going against the flow of diffusion. It can be used for larger particles, things that simply just can't fit through the membrane. And quite often, these are then utilizing ATP directly as an energy source, because you somehow need to add energy into the system to cause these to occur. Let's look at the sodium-potassium pump. Now, this is going to be an example of going against a concentration gradient. The cells in your muscular system, in your nervous system, they're going to have this resting membrane potential set. The way they do that is by having a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. So that's really great. When you open the channels up, sodium rushes in, potassium rushes out. But then you need to reset this gradient, which means you need to push the sodium out of the cell against its concentration gradient. You need to pull that potassium back in the cell against its concentration gradient. So on this diagram here, you can notice that the sodium is the orange ones and the potassium are more yellow. You can see how there's a high concentration of sodium outside here, where you have a high concentration of potassium inside the cell here. That's the normal homeostatic balance. When the cells are at rest, you have a high concentration of sodium outside, high concentration of potassium inside. But you need to be able to reset this. So the cells are constantly pushing these molecules against these concentration gradients. You can see here how you have the sodium is inside this sodium-potassium pump, this protein. What will end up happening is, as you move over, you can see how the sodium was released into the higher concentration side. That was accomplished by the addition of ATP. Adding ATP in was the energy that pushed those sodium molecules from the low to the high concentration, so against their concentration. And the same thing happens with potassium. ATP allows to go against its concentration gradients. We also have things called cotransporters. Now, the first example, to give you an idea, is called a unitransporter. It's not a cotransporter. It's one single particle, particle A, traveling through. Cotransporters are when you have two or more particles transporting through. So to see the difference, you can see one trap going through, that's a uniporter. But when we talk about these cotransporters, you have the symporter. That means both of these are going through together. Symport, both particles go through in the same direction. But then you have an antiporter. Antiporter is when they're going in opposite directions. One's coming in, one's going out. So they're going in the opposite directions. So some different types of transportation methods here. Again, these could be utilizing ATP as an energy source to push them in the opposite directions. Here's an example of the sodium glucose symporter. Now, we already mentioned how sodium is set up as a high and low concentration. We know you need an active transport to push the sodium against that concentration gradient. But when that concentration gradient is set, simply open the channel and the sodium wants to rush through. So the sodium itself is trying to get in, because you can see how there's a lot more sodium outside here compared to what's inside. So the outside has the higher concentration. The inside has the higher concentration of glucose molecules. There's more of them. Which means as the sodium comes into the cell, it's going to pull the glucose with it. So sodium has a very large gradient here. That large gradient means there's a lot of energy stored up to come in. It wants to enter the cell. So as it comes in, it'll drag a glucose molecule through this transport system, even though glucose is going against its concentration gradient. There's enough of a concentration gradient from sodium to pull it right along. So it's the idea of the sodium-glucose symporter, utilizing the energy stored in the sodium gradient to pull the glucose against its gradient. Then we have something called endocytosis. Endocytosis is a way of pulling particles in through the membrane. But you'll notice in this, all three of these are using some membrane-formed compartments. The first example, phagocytosis, it completely engulfs a particle and puts it into a vacuole. This is the idea of phagocytosis. The cell membrane engulfs and a lot of times then eats and destroys that foreign particle or whatever it's trying to get rid of. So the idea of phagocytosis. Next one is pinocytosis. Pinocytosis is almost like the cell's kind of drinking, it's pulling fluid in with ions as well. The membrane still is going to take a gulp of liquid here. It's going to close it off, and that's going to bring it inside the cell in a vesicle there. It's bringing fluid and particles, that whole kind of gulp of fluid there, into the cell. That's the idea of pinocytosis. Last one is receptor-mediated endocytosis. Receptor-mediated because you're going to see all these receptors are located here. Those receptors are particular for a certain molecule or particle. When that molecule or particle binds, it allows them to form this entire vesicle. Because they grab these things, the membrane forms around it, and then it pulls it into the cell. It's a way of bringing other components, particles, molecules in that cannot pass through the membrane. So different ways of endocytosis, taking in. But if you take in, you better be able to push out also. That's exocytosis. Exocytosis will take things that are inside the cell, form kind of this membrane structure around it, then open up to the outside. By opening up to the outside, those particles that were inside here can be released out to there. It's a way of getting rid of waste products or excess particles, but removing things from inside the cell. Exocytosis, going out. Again, both these are using ATP, endocytosis and exocytosis. Both utilizing ATP to move things across the membrane. So a little bit of a quick review from both passive and active transport. Remember, we had simple diffusion, facilitated diffusion, and osmosis, filtration. Those are all passive. You can see that the simple diffusion, facilitated diffusion, and osmosis were kinetic energy, while filtration used hydrostatic pressure there as its energy. The active transports of solutes, endocytosis, exocytosis, those are utilizing ATP. So we have different transport methods. And if you're wondering, why do I even care about all these? It's those transport methods, they're going to help move particles across the cells that will be important for every single system we're covering in the future. Because you have to move particles in and out to maintain homeostasis, allow for communications, and pretty much every organ in the body needs to be able to work this way. So these will come back in the future for how all these organ systems work. So a little bit about membrane transport.